conventional approaches just to evaluate community dynamics. So the idea of my talk was sort of to integrate the two approaches and try to find a balance on what can come out when you integrate, on one end, the use of stepwise slopes to evaluate food webs, and on the other hand, just the conventional approaches that we often use. And the primary findings from this approach clearly shows that each one approach gives you something that is slightly different from the other, but in a way that could be treated as complementary. And specifically, our assessment of community dynamics using conventional approaches highlighted the relative importance of seasonality when one evaluates the role of anthropogenic disturbances. On the other hand, when you evaluate different levels of disturbance using food webs, you tend to start you start to see areas where aspects that are more similar than others, or the nitty-gritties in terms of differences. So one could perceive that as using different lenses in trying to observe the same picture. So in a nutshell, that is what my talk is all about. Thank you.
Thank you very much, Wilbert. It's very interesting. I like the idea of using the two different approaches and then seeing how they can complement one another and make up for areas that are lacking maybe in one of the methods of the other. Thank you for that. Moving on, I would like to ask Lubabalu Morfu to please give us an overview of his work on the irrigation dams in the Eastern Cape. Luba, are you there? It might seem that Luba is not available. Okay, so I think we'll move on to the next speaker, and then maybe Luba will join us a bit later. From here, then, I think we move into KwaZulu-Natal, and I would please like to ask Matthew Bird to give us an overview on his work on the aquatic invertebrates in the Isimangaliso Wetland Park. So, Matthew, are you available?
I am here.
Great.
So what this work did is we went and sampled a freshwater lake on the coastal plain of KwaZulu, Isimangaliso Park, and no one's really ever looked at this lake, barely ever taken a water sample, looked at anything living in it, and we just wanted to have a look-see, and we wanted to also assess whether there is this estuarine relict fauna living in this freshwater lake, which is what we've seen, or what other people have seen in some of the other coastal lakes in KwaZulu-Natal that are freshwater, because most of these lakes were once connected to the ocean. And we sampled a wet season and a dry season back in 2013, 2014. We found most of the macroinvertebrate assemblage to be typical freshwater assemblage, and quite rich at that, quite diverse. But we also found abundant amphipods that are estuarine relics. So they're still sticking around, living in this lake, even though for thousands of years it has been fresh water. And quite interesting, a situation sort of like what we see with Lake Sabaya. And also two of these species that we recorded were actually our estuarine relic amphipods that have never been, to our knowledge, recorded in fresh water. So that was quite an interesting finding. And yeah, generally the lake seems to be very diverse. We do a descriptive comparison in the study with the number of taxa recorded from other lakes in the region, freshwater lakes, and find that although our lake is only about 1.7 kilometers squared, it's smaller than the other lakes we compared it with. It actually has higher biodiversity, and some of the other sampling programs have actually been more intensive than ours. So given given limited sampling effort, we found quite a remarkable biodiversity in this lake. And yeah, we just argue for the conservation of this lake, given that it is, luckily it's already in a pristine condition and it is in a conservation area. And lakes like this, we just argue must stay this way and make sure they stay this way and don't become polluted. These are also, given that coastal freshwater lakes are very rare in South Africa.
Awesome. Thank you, Matthew. It's very interesting. I actually didn't even know about this particular freshwater lake. So it's nice to see it highlighted as well and bringing to light more information on these not ignored, but not well-studied systems. Keeping with the aquatic macroinvertebrates, I would like to now ask Katlego Mahlope to tell us a bit about his research on the thermal tolerance of a beeted species from rivers in Gauteng. If you'd be so kind.
Hello, can you hear me?
Yes, I can hear you.
Okay, no, thanks. Well, I'm Katlego Mahlope, as said, from the University of Johannesburg. I did my project on the thermal tolerance of beeted sarasoni from Johannesburg streams. So now the reason for conducting this project study was due to the fact that there's absolutely nothing which has been documented when it comes to thermal tolerance on aquatic macroinvertebrates. And you realize that, in fact, in the Highveld, there's absolutely nothing which has been documented on thermal tolerance. But there's few studies which have been done in the Western Cape, Eastern Cape, as well as Mpumalanga and KZN. So now the aim of the study was basically to determine the degree of variation in thermal tolerance among populations from different streams and whether this is influenced by differences in thermal history. And in terms of the hypothesis, we hypothesized that populations inhabiting warmer streams were expected to have higher thermal tolerance than those that come from cool conditions. So now the collected individuals were exposed to LT50 technique, which measures the survival rate of species. So now you realize that the collected individuals were exposed to different effect temperatures over a period of time. So now in terms of the outcome of the project study, it was realized that the LT50 results mostly supported the hypothesis that thermal history is positively related with thermal tolerance. But there were a few inconsistencies when it comes to the results. So now in terms of the recommendation, it was suggested that some of the inconsistencies which might have occurred in the project study might have been due to multiple stresses such as pollution. And the fact that another thing which must be considered is that sampling was done only for 3 seasons. We couldn't do it for autumn season due to unforeseen circumstances. Yeah, so now in terms of the take-home message relating to the project study, I would say that thermal tolerance varies across different seasons and different streams. Yeah, thank you.
Great, thank you very much for that overview. It's really interesting to see some of the work on this, on the effects of different temperatures on some of these smaller invertebrates. Now I would like to move into the nature reserves of the Free State and ask Anke de Smit to please tell us a bit more about your work on the nematodes of the nature reserves there.
Yes, thank you, Dr. Lazan, and good afternoon, everybody. Can everybody hear me?
Yeah, yes, we can hear you.
Right, so I'm from the University of the Free State at the Department of Zoology and Entomology. And my presentation was about my master's study that was conducted between 2017 until January 2019 on nematodes that was found in aquatic habitats in 2 nature reserves in the Free State province. So just to give you a quick overview regarding the study, Nematodes are known as non-segmented roundworms, and they inhabit every known ecological niche on Earth. And they also regulate the turnover microbial communities, and thus they have a really important role in ecosystem functioning. And there are very little data available on free-living nematodes found in freshwater systems, especially in nature reserves in the Free State province. So therefore, it was decided to conduct a survey for my master's study on free-living and plant parasitic nematodes from both aquatic and terrestrial habitats occurring in Sudurang and Willem-Pretoria Nature Reserve. But for this presentation, I only talked about the nematodes from the aquatic habitats that was found during my study. And this is from a river habitat in Sudurang Nature Reserve and a dam habitat in Willem-Pretoria Nature Reserve. So the objective for the study was to map the biodiversity of nematode genera and to investigate the ecological diversity and characteristics of nematode fauna. And overall, the study showed that there were a total of 36 nematode genera belonging to 24 families that were collected at both reserves, and that bacteriivore nematodes were the dominant trophic group throughout the surveys in both reserves. There were some plant feeder nematodes, which were also present in the aquatic habitat, which could be due to vegetation occurring in the area where sampling took place. So the study provides a necessary baseline data for similar studies on nematodes from nature reserves in the Free State. It is also important to study free-living freshwater nematodes as they are seen as bioindicators in their surroundings. Thank you, everybody.
Great, thank you very much, Anke. I personally don't know the nematodes that well. I just know that they are found just about everywhere. So it's great to see that there's a little bit more work on the ones that aren't really studied. I would like to ask Anika Krier to please tell us a bit more about her research on the Cladocerans, also in the Free State province.
Okay, thank you. Can everyone hear me?
Yes, we can hear you.
Okay, good afternoon, everyone. I am a final year MSc student at the Department of Zoology and Entomology at the University of the Free State, and my MSc thesis focused on the taxonomy of Cladocera in the Free State in 6 different nature reserves. Between the 6 different water bodies that were sampled, Allemanskraal Dam was the most diverse, so Cladocerans present in this dam was the main focus for my presentation. Little taxonomic effort has been made in this region and has therefore led to several species being underdescribed or not described at all. The main goal was to focus on redescribing already known taxa in more detail to update already available keys. This was achieved by studying the morphology of all Cladocerans found in extensive detail. For Allemanskraal Dam, 10 species belonging to 8 genera and 5 families were identified. One of the species identified is a new distribution record for this region. The taxonomic composition of Cladocerans identified during this study demonstrates how high diversity can be in just one dam and how much taxonomic information can be obtained from one detailed study. So ultimately, it emphasizes the fact that morphological traits must be carefully studied in order to successfully, fully describe and distinguish species from each other. Thank you.
Thank you, Anika. Now, the Cladocerans can be a bit of a difficult topic in terms of identification, so it's impressive the work that you did and the great diversity that you found. It's really cool to see. Now we will move into research more on specific invertebrates, other specific invertebrates. Here, mosquitoes. So I would please like to first ask Gordian Mataba to please tell us about his work on mosquitoes, if you are there.
Yes, I'm here.
Okay, great. Thank you.
Okay, thank you too, and good afternoon, everybody. Hello, do you hear me?
Yes, we can hear you.
Okay, thank you. My talk, which I am presenting this conference, is about the PhD study which is ongoing, and we are investigating on different strategies to biologically control mosquitoes. We are meaning mosquito oviposition and the larval development. And actually, we are using mesocosm experiments. But before embarking to that, to those experiments, we studied with a field survey, where we actually wanted to see actually what is happening or what is actually existing in the field, meaning that we wanted to learn on which mosquito species and the macroinvertebrate species which are actually present in this region, and also wanted to know which are the main mosquito breeding habitats in this region. And the third, we wanted to know what actually controls the abundance of mosquitoes in this region. And here we included the effect of land use, to see how land use practices can either positively or negatively influence the abundance of mosquitoes. And this information was actually very important. Different mosquito strategies which can be applied in the region. So we sampled the temporary ponds, the small natural habitats, and the artificial anthropogenic habitats. And from the ponds, the primary findings, we found that from temporary ponds, the abundance of mosquito larvae were very, very few, meaning that few mosquitoes are utilizing temporary ponds as their breeding sites. And what kept that number quite low like that was the abundance of aquatic macroinvertebrates. Here I'm meaning the aquatic predators which prey on mosquitoes. So the take-home message from the ponds is that during mosquito control programs or activity, the ponds in the region should not be the habitats to be prioritized for any sort of larviciding. And if larviciding is necessary, then prioritizing temporary ponds for mosquito control would just be a waste of resource. And if, for example, that larviciding involves application of a chemical pesticide, then that will be unnecessary pollution of the ponds. And as we all know that these temporary ponds, especially in semi-arid regions, like in our study area, they are the main sources of water during wet seasons. So they provide water for livestock, wildlife, for domestic uses. But also, these ponds, they have very unique creatures which are endemic to temporary ponds. So if larviciding using a chemical pesticide will be done in these ponds, then it will endanger the survival of these aquatic creatures. And from the small natural aquatic habitats, we found that these were the main breeding sites for the anopheles mosquitoes. And therefore, these are the ones which should be prioritized during mosquito control. And particularly, we saw that we found that the level of turbidity, of the water turbidity and the proximity of the habitats to human habitation were the major factors which were determining the abundance of anopheles larvae in these mosquito habitats. And from the artificial container habitats, we found that these were the major breeding sites for the Aedes aegypti and the Culex quinquefasciatus mosquitoes. And therefore, this conveys a message that these are the ones which should also be prioritized for the control of Aedes aegypti and the Culex quinquefasciatus, particularly by covering these containers and the frequent emptying of these containers, for example, frequent emptying and the refilling, because covering of the containers will prevent access of the gravity mosquitoes to lay eggs in the containers, and the frequent emptying and refilling will interrupt the mosquito development cycle. So generally, in this region, we came up with a general conclusion that land use actually the effect of land use, the chemicals from the agricultural areas and different land use practices actually are not influencing positively or negatively the abundance of mosquito larvae, but other factors such as type of habitat and biotic factors such as macroinvertebrates are the ones which actually positively or negatively limit the abundance of mosquitoes. This is all. Thank you for your attention.
Great, thank you very much. Research on the temporary wetlands and ponds and so on is quite close to my heart as well. So I always enjoy learning a bit more about the research that's going on there. Finally, I would like to end this the particular session's talk by asking Baledi Buxton to please tell us about the research on the knowledge, perceptions, and practices of people also surrounding mosquitoes.
Thank you very much. I believe you can hear me.
Yes, I can.
Yes. My name is Baledi Buxton. I just graduated with a PhD from Botswana International University in Science and Technology a couple of months ago. And the work I'm presenting on today was actually a survey aimed at assessing community knowledge on mosquito biology and ecology. That is how much they know about mosquitoes and their ecology. And the ultimate effort here was to manage mosquito population and associated emerging and re-emerging infections within the household level. And the survey here was carried out mainly because in normal scenarios, expensive control intervention strategies are used prior to assessing the knowledge gaps of the affected communities. That is the deployment of expensive chemicals or molecular approaches without touching base with the affected people on ground. The survey was carried out in two non-malaria endemic sub-districts in Botswana and one malaria endemic district. That is to strike a balance to see if the knowledge would otherwise be the same or would there be any significant difference when testing all these knowledge gaps. And overall, there was limited knowledge across districts, regardless of the malaria endemicity status. In other words, it was just the same. The knowledge limitation was just the same across all the districts. And for the interests of this conference, the majority of respondents did not know or have never seen the aquatic life stages of mosquitoes. That is the egg, the larvae, and the pupae. And this is very interesting because it makes it difficult to associate mosquitoes with water and hence the control interventions. And in short here, I could say the take-home message is that let us and the communities around us strengthen our knowledge in mosquito biology and ecology, because this is the fundamental step towards the control of problematic mosquito species, especially in the neglected aquatic habitats. This is because there are many alternative ways in which we can control mosquitoes in aquatic habitats, maybe the use of aquatic predators as an inherent ecosystem service, or maybe management of those water holding containers that probably might serve as breeding sites for mosquitoes. And also because from this study, we realized that much of the control was skewed towards the adults, which of course they know, but nothing was done on the aquatic life stages. So the main message here is that the mosquito life stages need to somehow be managed from those aquatic habitats. With that, ladies and gentlemen, I say thank you.
Great, thank you very much. I think your talk and the previous one actually go quite well hand in hand to just show how people don't understand or know that well about the larvae, etc. And if there's better education, then people might be able to learn more, not just about the adult stages, but the larval stages as well, and how they can be controlled better. So that's really nice to see. I'd just like to again ask maybe if Lubabalu is here. Otherwise, we'll move on to the question and answer part of the session. Okay, I don't think Luba is here with us right now. So with that, if anyone would like to ask any specific questions, now is the time to do that. Oh, I see Wynand's hand is up. Wynand, if you would like to ask your question.
Thank you, Lizanne. I think it's a question for Matthew on the work there in the lakes in northern KwaZulu-Natal, and it's more of a comment probably. And it's great that he's doing this work, or he did the work, because there's many of these lakes and larger wetland systems where there's absolutely no information about the macroinvertebrates and the distribution of these taxa and which ones occur, and it's really a big research need that more of these smaller wetlands, lakes need to be surveyed for macroinvertebrates all around the country, not just northern KwaZulu-Natal, I think.
Yeah, thanks for the comment, Wynand. The lakes in the two clusters of lakes, basically, the wilderness area and the garden route, and then the lakes of KwaZulu-Natal, which extend basically from Durban up into southern Mozambique. And it's most of the freshwater lakes are actually concentrated in this KwaZulu region, and the wilderness lakes, as far as I know, are mostly brackish or estuarine, except for Groenvlei. I think there's one truly freshwater lake there. So if you consider that basically in terms of freshwater lakes, almost all of them are in northern KwaZulu. And the number I had from a paper there in my talk was something like 27 of them in the whole country, and most of them are less than like a kilometer squared. I think only 18 are bigger than a kilometer squared. So it's a really scarce habitat. We just don't have many of these. And maybe that is part of why there's not a big research effort on these lakes. Maybe we just tend to focus on the systems that we're so used to seeing and that are commonly being polluted and whatnot around the country, our typical pans and river systems. So yeah, these lakes are just not very well known and pretty much neglected lately.
Okay, thank you, Matthew, and thanks for the question as well, Wynand. I see there's a question from Herman, if you would like to.
Yes, thanks, Lizanne. Matthew, it's Victor Wepner speaking. What was interesting was that, and as you also pointed out, that one of the surprising results that you got was the presence of Granidarella bonnieroides, together with Granidarella lignorum. Do you perhaps know why Bonnieroides was there as well? Because I know from sampling that area we used to get that in marine dominated systems.
So as I mentioned, so basically this lake was an estuary. I don't know the scientific details, but thousands of years ago, I think the Pleistocene transgression. But it's I couldn't find a publication saying it was an estuary, but most of the lakes in that area are estuarine relics. So these guys have been hanging around since then, and Bonnieroides is not a typical estuarine relic, but it is found in, I think it's found, I can't remember what exactly I read, but it's been reported from St. Lucia and some of these estuarine systems in the area. And so it's sticking around in fresh water since closure of this estuarine system, but it really is a weird one, because in the other coastal freshwater lakes, The Granularia has only really been Lignorum, that species, which is, the Lignorum is quite commonly found in freshwater systems that were once estuarine. It's not unusual. But this Bonnieroides was a weird one. And we did confirm the ID. Yeah.
It is quite weird because it's usually found in very recent marine-like systems. So it was quite dominant in the more shallow, soft bottom habitats of, for example, Richards Bay Harbor. So it's quite interesting that this recent marine species is there. Perhaps it's got to do with it not being so or separated from the marine environment for as long as the other coastal lakes. Who knows?
Yeah, I actually don't have specific information on that. I would love to chat to a sort of geologist or someone who could really explain. You know, I don't know if this particular, I couldn't find specific geological info on this particular lake, but there's quite detailed information on a few of the other coastal lakes in the area. Bonnieroides has been recorded at Lake St. Lucia in quite fresh conditions, but temporarily fresh. You know, when you have these sort of high freshwater input events and rainfall and whatnot. And there's a paper an old paper where they said they were also surprised to find it under fresh conditions, but that's more like temporarily fresh. So it's very weird that it was in a permanently fresh system at the moment.
Great, thanks, Matthew, and thanks, Prof. Vic, for the question there. Do we have any other specific questions? So I see there's a comment, Matthew, from Ryan Wasserman, who just says, nice talk. Would be cool to look at the genetic isolation. It may inform on when this population was isolated. And actually that links to a question that I wanted to ask as well, because I was wondering myself, where exactly would these estuarine species be coming from? Like, do you think they were just isolated when it became, and then, or is there somehow, are they being introduced from somewhere else? I mean, I can't imagine. So that I also agree the genetics would be pretty interesting to see.
Yeah, to my knowledge, they just stick around. They didn't come. They stick around from when it was an estuary. So they've been in the system the whole time. That's what's been found for Sabaya and some of those other freshwater lakes. Okay,
cool. Thanks.
But yeah, one last comment is it would be definitely interesting to look at genetic techniques to actually figure out how long they've been there, and you know how it will work. You could get more specific information using the genetics for sure.
Okay, cool. Do we have any other questions or comments from the audience? I would actually like to ask Anika a question. Just out of interest, like the diversity of the cladocerans that you found, you said it was quite high, but how does it compare to the diversity of maybe other systems? Is it very similar? Is it much greater than those? Or yes, I just wanted to, in terms of comparison with any other ecosystems.
Yes, so like I mentioned, for my whole thesis, I focused on 6 different dams. And I mentioned that Allemanskraal Dam was the one with the highest diversity, but like diversity in other dams were never lacking. I think the one with the lowest number of species was 4, which is actually very high considering that we only sampled once in every dam. So the diversity, the pattern of high diversity actually continues through all of these systems. And I think if you would expand the study to more than just 6 dams and maybe to other regions, a similar trend would be followed as well.
Okay, great, thank you. And then just in terms of I don't know if you know whether any of these particular species are sensitive to environmental changes or pollutants or something like that?
Yes, definitely. So environmental changes which can include maybe like stresses like the water levels lowering or predators introduced to the waters that can actually induce several like changes in the organisms, like cyclomorphosis in some specimens that was actually found in other environments. And then if you would maybe get an environmental stress like the drying of a dam, they actually have a way to counteract this by forming these resting eggs that would, and they can survive in the sediment for years even. So when the conditions return to favorable conditions, they can actually just repopulate the waters by themselves again.
Okay, cool. Thank you for that. Does anybody else have any particular questions or comments for our speakers? Oh, I see there's a hand from Candace Janssen Rensburg, so you can ask a question.
Hi, Anika. I just wanted to find out from you, what is your opinion? Do you think if you sampled, say now we didn't sample in August last year and we sampled in April, for instance, do you think we would have found different results?
Yes, there would definitely be a difference between like seasonal variations, because there would obviously be more reproductive success in the warmer months, because the colder waters are definitely an environmental stress on them. But I think the study proved that the diversity still wasn't low, but maybe this wouldn't necessarily have an influence on the number of different species that we found, but maybe just a difference on the amount of individuals that we found.
Okay, thanks.
Okay. All right. Thank you very much for your question there, Candace, and thank you to all the speakers for giving us a bit of a background and more information on your topics. If there are no other specific questions or comments at this time, I would like to close this session. Thanks to everyone who participated, and thanks for all the interesting information you gave us. We will take a 10-minute comfort break, and then we can start the next session at just before 2, or at 2. Is that 2? Yes. So thank you very much, and we'll see everyone, see everyone again at 2 for the next session. Thank you.